Hello to all of you. I would like to discuss the SubCell Lab with you guys, and I'm going to go over the important parts of the lab with you so that I can make you understand what this lab is about. And I have already provided you with the data for this lab. So basically, making use of the data, you are going to answer the questions and you are going to submit your assignment on D12. The folder is already created. So basically, and I also have provided you with the PowerPoint. You can go over that, which explains you more about this technique. So basically, subcellular fractionation is a lab in which basically we break the cell and then we different organelles of the cell. So we use a method in this technique called differential centrifugation. And what is that method is basically, first of all, we break the cell. So basically in this lab, if we would have done in the lab, you would have seen that we take cauliflower cells and we basically take them in the mortar and pestle. We just grind them. We have a grinding buffer which we use in the lab. So we grind them, the cells break, the lysate is formed, and then we filter it, and then we collect the filtrate. We pass it through the cheesecloth, we collect the filtrate, and then we take that filtrate and then we spin it down under a low speed so that the heavy organelle, which is nucleus, it gets it basically settles down as a pellet, and then we take the supernatant, we spin that supernatant at a higher speed, because nucleus is a very large organelle, so it settles down at a low speed, and the other organelles like mitochondria, they will settle down as a pellet when we use a very high speed. So basically, it's a method of using differential centrifugation by which we increase the speed of the centrifugation, and we settle down these different organelles. So when in the first step is using low speed, so the big organelle, the high density organelle like nucleus, it settles down, and then we collect the supernatant, and in that supernatant there is no nucleus. We take that supernatant and we use a high speed, and the organelle like mitochondria and all that, then they settle down. So basically, this is the overall technique. So basically, in this technique, which is done in the lab, we isolate nucleus and the mitochondria. So we basically, for the nucleus, we look for the nucleus in the P1 fraction, and also other fractions also you can see in the protocol. I'm going to go quickly over the things. And then we use a staining technique called, we use a dye called Azure C, and with that dye we stain the nucleus. So that picture is shown in the PowerPoint, you can see. So basically what I'm going to focus on is how to detect the presence of mitochondria in different fractions. So basically we collect the filtrate, then we have different fractions like P1, S1, P2 and S2. So we take the filtrate, we spin it down, the material which deposits at the bottom, that is P1 pellet, and the supernatant which is there, that we call it S1, and then we take the S1 and we spin it down at a very high speed, and then we get some material which gets deposited and we call it P2, and the supernatant which is there, that we call supernatant 2. So we have 5 fractions. We have filtrate, we have P1, we have P2, we have S1, we have S2. So in these fractions we look for the presence of nucleus, we look for the presence of nucleus in filtrate, we look for the presence of nucleus in P1, and also we look for S1. And basically we expect the mitochondria to be in the P2, but we look for the presence of mitochondria in all these fractions, and that is mainly the lab is focused on for looking for the presence of mitochondria. So how do we detect the presence of mitochondria in the different fractions? For that we make use of the enzyme called SDH, which is succinate dehydrogenase enzyme, SDH. So basically this is an enzyme which is present in the inner membrane of mitochondria. So we make use of this enzyme, and we try to look for the presence of mitochondria in the different fractions. So this enzyme basically catalyzes a reaction which is the conversion of succinate to fumarate. So this is a one-off reaction in the Krebs cycle. So this enzyme, so when succinate is converted into fumarate, at this time succinate gets oxidized, which means the electrons are stripped from succinate, and they are in this step FAD, which is a coenzyme, gets converted into FADH2. So and then this FADH2, which is a electron carrier, this feeds the electrons into the electron transport chain, and in the electron transport chain there are four complexes, and eventually these electrons are accepted by oxygen, which is the final electron acceptor. So how to detect the presence of this enzyme? We make use of a system. In this experiment we make use of sodium azide, and sodium azide is basically the electron transport chain blocker. Basically the electrons, they flow through these complexes and they reach the oxygen eventually. So we are going to use, when we do the experiment in the lab, we use sodium azide, and it basically stops the flow of these electrons through these electron transport chain, and it blocks that. And in this experiment we use an artificial electron acceptor called DCIP. So when DCIP accepts the electrons, it gets reduced. So DCIP is blue in color. So when DCIP gets reduced, so DCIP is blue in color. When DCIP gets reduced, it becomes colorless. So there is a change in the color. There's a change in the color from blue to colorless. So that is what we are going to detect. So if this enzyme is there, which means mitochondria is there. If this enzyme is there, mitochondria is there, which means then there will be oxidation of succinate to fumarate. There will be oxidation of succinate to fumarate. And if mitochondria is there, this enzyme will be there, this reduction will be taking place. If this reduction is taking, this oxidation will be taking place, succinate to fumarate, and if and reduction of FAD is taking place, FAD is getting converted to FADH2, and it is going to feed the electron transport chain. But because we are using sodium azide, which is going to block the electron transport chain, and we are also using artificial electron acceptor DCIP, it will be receiving these electrons. It will be receiving these electrons, and it will be getting from blue to colorless. So if we are getting the change of color, if we are getting the change of color from blue to colorless, if we are getting the change in color from blue to colorless, it means that in that fraction this enzyme is there. If this enzyme is there, mitochondria is there. So making use of this system, we basically try to detect the presence of mitochondria. And in this experiment, we basically use various controls. So we use a control, malonate control. So malonate is basically a competitive inhibitor for this enzyme STH. So it competes with succinate. So we use that. It's one of our control. Then in another control we use sodium azide. We basically in this control we do not use sodium azide, and we like to see if the what happens to the reduction of DCIP. And the last control we will be not using succinate. So here is a table to understand all this, and I can explain you from this table. I have already explained you overall protocol. So this is the table. This is the table. This is the way this experiment is done. So basically we would try to see for the reduction in the color in all the fractions. So we use filtrate, we use S1, we use P1, we use S2, and we use P2. And for all of these fractions we also use their blanks. So blank is the one which we use actually when we do this experiment. We have to look for the change in color using the spectrophotometer, and we look at 600. So for every fraction we need a blank, because we use the wavelength 600. We use the wavelength 600, and for the calibration of the spectronic for every fraction we need to have a blank. So so we need to have a blank for every fraction. So what is a blank? Blank is something in which we don't use the DCIP. So this is used for the calibration. In all the other fractions, what do we add? We add the mannitol assay buffer, we add azide, we add DCIP, we add succinate, and we add the respective fraction. And once the fraction is added, the time point starts. So in this experiment, this is the plan which is written here. I can show this is given in the table. I don't want to go over each and everything. So basically the basic plan is, in every fraction, for every fraction we have a blank in which we don't use DCIP. We use the assay buffer, we use azide, we use succinate, and we use also DCIP. We use DCIP, azide, succinate, and we add the respective fractions. Okay. So this is, and also we have used some controls here. As I told you, we use malonate, which is a comparative inhibitor, and we use azide control, which means in this we are not using azide, and succinate control, which means we are not using succinate here. And in all these controls, we use the fraction which we use is the P2. And what we do here is we make the use of blank and we calibrate the spectronic, making use of the blank. And then in all of these fractions, in all of these tubes, basically we make them these mixtures in the cuvettes. We first of all take the reading at 0 minute, and then we take the reading at 3 minutes. So once you add the fraction, your time point starts, and after 3 minutes you look for if there is a reduction in the color. So if the reduction of color takes place, which means this enzyme is there. If this enzyme is there, mitochondria is there. And so in the controls we expect the reduction to be less, because we expect the system not to work in the controls. So, and this is the formula. This is a very important formula you need to know. I have already given you the data for the readings of the OD at 3 minute and 0 minute for every fraction, and I have also given you the change in OD which takes place in 3 minutes. So this is a formula by which you can first of all find out the concentration of DCIP which is reduced. The concentration of DCIP which is reduced, you can figure out by taking the change in the OD divided by the extinction coefficient of DCIP, which is given here, and its value is 21, and this value is in millimeters. So millimeters divide by the so this is the formula which is given here. So the concentration of the DCIP, the reduced DCIP we get, that value comes in millimolars, and from that we can calculate the amount of DCIP which is reduced. The amount of DCIP reduced can be calculated by taking the value of concentration of DCIP which is reduced and multiplying it with the volume, and the volume is 2 ml. And this value which we calculate from the concentration above, it should come in millimeters. It's very important. It should come in millimoles. Okay, so the value comes in millimoles. Here it is written micromoles. So if you there is a choice. If you want to keep the value in millimolars, you can keep it in millimolars. But if, as it is written here, if you want to convert it into micromolars, you have to multiply by 1000. You have to multiply by 1000. And how to calculate the velocity or the rate of DCIP reduction? You have to divide by the reaction time. You have to divide by the reaction time, and the reaction time is 3 minutes. Okay, so the volume is 2 ml and the reaction time is 3 minutes. So you divide the moles of you divide the amount of DCIP reduced by the reaction time. That gives you the rate of DCIP reduction. So if you want to get the answer, if you don't want to change anything, so the answer will be millimoles per minute. But if you want to get it into micromoles, then you have to multiply by 1000. So this is basically what you have to do, and the data is already given to you, and these are 5 questions. You can answer these questions. If you have any question, you can send me email, or I can also have a virtual meeting with you guys on D12. But if you want to have that, you have to let me know 1 or 2 days before, so that I can set up a meeting there. Okay guys, that's all I wanted to explain you about the lab. I hope it's clear.